All right, in this video we're going to talk about some of the signals that are involved in the cell cycle. So in order to control the cell cycle, there are various signals that the cell produces or produced by other cells that affect the cell. One of the main groups of cell signals that are involved in cell cycle control are called cyclins. Cyclins are regulatory proteins that regulate when the cell moves from one stage of the cell cycle to the next. So as the cell does the things it needs to do to get ready to move to the next step, the cyclins get produced and their levels are going to go up, and then the cell's going to move on and the levels are going to drop off. There's another group of proteins or of chemicals that are involved in cell cycle signaling called cyclin-dependent kinases or CDKs. So these are enzymes. Kinase ends in ASE. So they phosphorylate various cellular proteins, they add energy, and they activate or inactivate various proteins. So when the cell has done all the things it needs to do to move on to the next steps, the cyclins build up. The cyclins combine with the CDKs to form the CDK cyclin complex, and they activate the CDK. And this triggers the movement of the cell through the various stages of the cell cycle. So in the 70s and 80s, there were several different researchers that worked on figuring how all this worked out. Hartwell kind of was looking at the checkpoints, Hunt was looking at CDKs, and Nurse looking at cyclins, and kind of put all their work together. And they looked at how CDKs and cyclins trigger the different stages of the cell cycle. So again, at the various checkpoints, there are various cyclins that have to be produced in order for the cell to move past that checkpoint. So growth factors, the nutritional state of the cell, the size of the cell, all play a role in determining when the cell is ready to go from the G1 to the S phase of the cell cycle. So when the CDKs are activated by the G1 cyclin, the cell is able to move on to the next step. At the G2 mitosis checkpoint, make sure that the DNA has been replicated, that it's all been copied, it's been copied correctly, and then the cyclins are going to be produced. It's called the G2 and mitosis promoting factor is the name of the complex. And it causes the cell to go through mitosis. And then at the spindle checkpoint, making sure the chromosomes are all attached at the metaphase plate. They're lined up. There's a spindle fiber attached to each of the chromosomes, and it's ready to go through the various stages of mitosis. And then the cell goes through cytokinesis after mitosis. So CDK cyclins drive cells from one phase to the next. In order to make sure the cell goes through the cell cycle properly and that everything's been done before it moves on to the next stage, we have this regulatory mechanism. So because this is such a key aspect of life, the genes for these regulatory proteins has been highly conserved. In other words, they haven't changed much throughout the history of life through evolution. They're all pretty much the same. So there are various external signals that can cause cells to divide as well. So CDK, cyclins, all of that stuff, that's internal factors. External factors are called growth factors, and they're coordinated between cells. So these are proteins that are released by one body cell that stimulates other cells to divide. They play a role in what we call density-dependent inhibition. Normally when cells start to reproduce and they kind of bump into other cells, as their density gets up to a certain level, they stop dividing. Now this is where cancer and tumors lose this density dependent inhibition. So the cells bind, a bit of growth factor is not enough activator left to trigger division of any one cell. There's something called anchorage dependence. To divide, cells must be attached to something. They can't be just floating around in some type of fluid. So there are touch sensor receptors that make sure the cell is attached to something before it starts dividing. So here's how kind of growth factors work. You've got a signal coming from somewhere else in the body. It binds to a receptor on the surface of the cell. It goes through that protein kinase cascade, that cell signaling, which ends up triggering cell division at some point. So an example of growth factor that we've studied quite a bit is platelet-derived growth factors. Platelets are cells that play a role in causing blood clots. So when this platelet-derived growth factor binds to the cell receptors, it stimulates cell division in the connective tissue and starts the healing process. Erythropoietin is another hormone that plays a role in making red blood cells. This is what Lance Armstrong got in trouble for, lost all of his Tour de France medals. A lot of cyclists have gotten in trouble for blood doping.